I'm happy to be with you today to share with you our work on copy number variation in the Tunisian population. So my talk will begins with an introduction on CNVs and followed by the results of our investigation of this kind of variation in the Tunisian population. So by exploring the Human Genome Project, the International HubMap Project, and the 1000 Genome Project, a lot of genomic sequence variation has been identified and compiled using a variety of techniques, including recently next-generation sequencing. The major form of genomic variation are shown in this figure. This includes SNPs, which are qualitative in nature, as they affect a single nucleotide only, as well as a family of genomic changes, collectively called the structural variation, because they affect the dosage or the copy number of a particular genomic sequence. The structural variation include duplications, deletions, as well as inversions, and range from small deletions and duplication to large genomic variation that involve the entire chromosome. It has been estimated that roughly 5% of the human genome is structurally variable. The first association of a CNV with a phenotype was the case of the bar-I phenotype in Drosophila melanogaster due to duplication of the bar gene. And after the human karyotype was established, it was possible to report chromosomal aberrations that were visible at the microscope in the human genome starting from the 60s, when cytogeneticists were able to identify the molecular background of many genomic disorders, including the Cri-du-Chat syndrome due to a partial deletion of the short arm of the chromosome 5. And for decades, and due to low resolution of cytogenetic methods, only large chromosomal and pathogenic chromosomal aberration were possible to identify that involve many genes and having adverse phenotypes. The concept of copy number variation arises and originates recently to describe genomic differences between individuals that were thought first as rare mutational events and shown later that could affect and could occur in normal individuals. So earlier studies identified CNVs and defined CNVs as segments larger than 1 kb, followed by 1,000 nucleotides, and recently a CNV is a segment larger than 50 nucleotides to several megabases. The experimental methods for identification of CNVs include a hybridization approach like CGH arrays and SNP microarrays, and recently next-generation sequencing that led to the exponential identification and characterization of structural variations. In addition, computational methods have been developed and integrated to accurately characterize structural variation and their content. In general, we have 2 copies of a gene in our cells, and due to several factors such as homologous repeats, segmental duplication, and non-allelic homologous recombination, the copy number may vary between genomes and leading to deletions and duplications. So when the 2 copies of a gene is deleted, we have a homozygous deletion, and this is the copy number state of 0. When 1 copy is deleted, we have a heterozygous deletion and this is the copy number state of 1. And when a copy is added along the two copies, this is a duplication and the copy number state is 3. And when two copies are added, this is also duplication and the copy number state is 4. CNVs could be benign and could also alter phenotypes using a variety of mechanisms, including dosage sensitivity of a gene within a CNV. And in this situation, the gene is called haploinsufficient, meaning it is intolerant to heterozygous loss of function. Duplication may also involve triple sensitive genes, and this has been evidenced when duplication and quadruplication of a particular gene, dosage gene, led to an exacerbate phenotype. In addition, a CNV could encompass 2 or more sensitive genes with overlapping phenotypes, and in this situation, the CNV is called contiguous gene deletion syndrome. In the situation of gene interruption and gene fusion, the direct interruption of a gene by a CNV could lead to the inactivation and loss of function of a gene. And finally, a CNV could affect the non-coding regulatory element, either upstream gene or downstream disease-associated gene. And in this situation, there is an alteration of the phenotype, and deletion and duplication or inversion may be involved. Biallelic CNVs could cause diseases either as homozygous deletions in consanguineous population or as compound heterozygous deletions. In addition, the combination of a CNV with a single nucleotide variant could be of clinical relevance. Indeed, a deletion could unmask recessive coding or non-coding variants, thus leading to a disease or phenotype when inherited in trans. At one extreme, CNVs could lead to diseases such as microduplication and microdeletion syndrome, like those with intellectual disability, like DiGeorge syndrome, Prader-Willi and Genman syndrome. And also, at the other extreme, CNVs could occur in normal individuals and have no known phenotypic effect. And between the two extremes, CNVs could lead to common benign traits like color blindness, infertility, hypertension, and also could predispose or could protect against diseases, such is the case of the copy number variation in the CCL3L1 gene that is associated to a lower risk to HIV infection in the African population. So CNVs could lead to dominant diseases, and in this situation, the CNVs are in general absent in the healthy population, and they could also lead to recessive and complex diseases, and in this situation, CNVs are present in the healthy population. The first genome-wide study of CNVs was performed on 217 HapMap healthy individuals from 3 different populations, and this study had allowed the identification of 1,447 CNVs across the genome. And the first CNV map was established and highlights the ubiquity of CNVs in the human genome. The functional annotation of the genes overlapped by CNVs using gene ontology and different pathway showed that the functions enriched for such diseases include immune response, xenobiotic drug and steroid metabolism, and starch and sucrose metabolisms. A more recent reference map for structural variation has been recently proposed, and this study included more than 40,000 genomes across diverse global population. The study also and this map include more than 400,000 structural variation, including CNVs, inversion, insertion, and translocation, with a median of more than 7,000 structural variation per genome. These structural variation are responsible for about 30% of all rare protein truncating events per genome, and confirm also the ubiquity of such structural variation in the human genome. The distribution of structural variation matched the expectation based on demographic history, human history. In addition, it was established that the African and African American samples showed the greatest diversity with more than 8,000 structural variation per genome, whereas the European samples showed the less diversity. Regarding the homozygosity, it has been shown that the East Asian populations are featured with the highest levels of homozygosity. The CNV map proposed by Zareh et al. In 2011 on healthy individuals showed that duplications and deletions overlapped with exon of genes with no phenotypic effect in majority, but also with homing genes responsible of diseases in humans, as well as essential genes for which homozygous mutations lead to embryonic and neonatal lethalities. The recent CNV structural variation map showed that more than 50,000 common structural variation were in strong linkage disequilibrium with at least one short variance, of which 50% matched a reported association from the GWAS catalog. It was also possible to calculate the CNV carrier frequency for 49 genomic disorders in the healthy individuals with no known neuropsychiatric diseases. This study, the recent study, also showed that there is no differences in the frequency of the CNV carriers of genomic disorders between populations, except with the exception of a duplication of a gene. In addition, it has been shown that less than 1% of samples carried a very rare structural variation, resulting in loss of function of a gene for which incidental findings are clinically actionable. In addition, more than 7% of individuals were heterozygous carrier of rare loss of function structural variation in known recessive developmental disorders gene. And finally, 4% of the general population carried at least one very large rare autosomal structural variation. And these results highlight the complexity of such structural variation and the challenge to interpret their pathogenicity or not in the human genome. So studies focusing on the characterization of copy number variation have been performed on different populations around the world, but they still unavailable in the Arab world, except for one studies on the Qatari population. And for this reason, we performed the first genome-wide study of CNVs in the Tunisian population. So the Tunisian population is a North African population of 11 million inhabitants. The native background is Berber, and the present and the genetic properties of the present population are shaped by the multiple migratory waves and invasions of different allogenic population and ethnic groups, mainly from the Middle East and Europe. And phylogenetic analysis showed the mosaic structure of the Tunisian population. Genetic diseases in Tunisia represent a real health burden with more than 500 genetic diseases identified. And the spectrum of genetic diseases is shaped by the high rates of consanguinity and endogamy, as well as by the historical events. Indeed, this spectrum is characterized by the high frequency of autosomal recessive diseases. We also reported that the three major disease groups were congenital malformation, metabolic diseases, and neurological diseases. In addition, due to the high rates of consanguinity, more than one genetic diseases could be expressed in one individual or in different members of the same family, and this phenomenon is called comorbidity. We also assessed the mutatome, which is the spectrum of germinal mutation, to more than 800 mutations, including founder mutation. But the CNV varium was unavailable and uncharacterized until the study, and we believe that CNVs could play a key role in the complex diseases, traits, or genomic disorders. So for this study, 135 Tunisian healthy individuals from different parts of the country were genotyped using the Affymetrix SNP array. So we recall that these individuals were healthy and free from any genomic disorders. CNVs was used to call CNVs. And after applying quality filters on samples, we kept only 102 individuals. We also applied quality control filters on CNVs by removing CNVs on telomeric, centromeric, and immunologic regions, and we kept only CNVs larger than 1 kb and shorter than 3 megabase. We also used another software, which is Birdsuite, with its two components, Bird's Eye and Canary, to call CNVs on these 102 individuals, and we applied the same quality control filters on CNVs on its output. So we merged the CNVs generated by PenCNV and Birdsuite to generate and build our global CNV map with 1,083 CNVs that were subsequently subject to descriptive statistic functional analysis and linkage disequilibrium analysis. So PenCNV outputs approximately 4,000 CNV on these 102 individuals, which means we have CNV carrier rates of 75%. The average number of CNV per individual was 39, and the number of CNV ranged from 17 to 63. Bird's Eye outputs more CNVs than PenCNV, reaching more than 300 CNVs per individual. In both situations, we genotyped more deletions than duplications. So we merged the individual CNVs into CNV region. So we get 751 CNVR from the PEN CNV output, and 1,263 CNV region from the bird's eye output. We identify three types of segments: segments with only deletions, segments with only duplications, and segments with both deletions and duplications that we call the mixed loci, and reflect the recurrence of CNVs at these locus. In both outputs, the genome coverage by CNVR were greater than 60 megabases. In addition, PEN CNV tend to identify larger segments than bird's eye, and in both outputs, we get duplications that are larger than deletions. So as CNV detection algorithms are plugged usually with high false positive rates, and as the two algorithms show different performance, we looked for overlapping segments between the two outputs as an in silico and concordance validation step. So we identified that 78% of the CNVR generated by PEN CNV overlapped with CNVR region generated by bird's eye. The frequency of this validated CNV regions ranged from singleton, meaning identified in one individual, to almost completely polymorphic. The average size was 19 kb. The validated CNV region were identified on all the autosomes, and we highlighted that the heterozygous deletions were the most common type of segments. The third algorithm that we used was Canary, and with Canary we genotyped more than 1,200 copy number polymorphism loci that were previously identified in HapMap individuals and having a frequency greater. So we get that more than 50% of these CNP were allelic, meaning that their copy number state was different from the neutral copy number state of 2. The frequency ranged from 1% to completely polymorphic. Regarding the size, CNPs were shorter than the CNV region, as the longest segments don't exceed 500 kilobases. We also identified three types of segment, those with only deletions, duplication, and mixed loci. All of the CNP identified on the different autosomes, and we also identified that heterozygous deletions were the most prevalent, followed by the homozygous deletion. So we merged the output of PEN CNV and the Canary algorithm to generate our CNV, the global CNV map. We also removed the false positive segments, and these false positive segments are in fact segments with frequency more than higher than 90%, and that were absent from the 1000 Genome Project and NOMAD project. So our global CNV map include 1,083 CNVs. The frequency ranged from 1% to 100%. We identified also the 3 types of events that were on the different autosomes. The average size of the CNV was 56 kilobase, and the genome coverage was 61 megabases. The duplication were larger than deletions, and the majority of the segments were shorter as their size doesn't exceed 60 kb. The heterozygous deletion were also the most prevalent. Nearly half of these CNVs overlapped with 1,018 RefSeq genes, of which half are protein coding genes. The genic CNVs encompass coding and functional elements, several disease genes, drug metabolizing genes that might contribute to the burden of chronic diseases. We also showed that the deletion spanning genes were more frequent. In addition, we found that the homozygous deletions are significantly gene poorer as it was previously shown. However, we found that rare CNVs seem not to harbor more genes than common events, and this result is in opposition with previous study that show indeed that rare CNV seem to harbor more genes than common events. So we didn't find such a relationship, maybe because of the modest size of our sample that underpowered the discovery of such a pattern. So in order to identify novel CNVs, we queried several structural databases, including DGV, 1000 Genome Project, NOMAD, DBAR, as well as datasets generated using next generation sequencing. So we looked for 50% of overlapping segments with the segments of these databases. So if a segments of our dataset overlapped with one segment of these datasets, then the CNV is called known. So we identified only seven copy number variation that were absent from these datasets. Among them, 6 were duplication and 1 deletion. All of them were singleton, and their size ranged from 7 kb to 700 kb. Among the duplication, so all the duplication overlapped with genes, with 20 genes, and among the RefSeq genes, only 2 are shown to be associated with diseases, namely congenital disorders of glycosylation type 2 and lung cancer. So due to the modest size of our sample, it is hard to confirm if these events are true rare events or not. We also noticed an increased burden of genic CNVs in KEGG pathways involved in infectious susceptibility disease. And among these genes, we have the KIR3DL1 gene, which encodes for a killer cell immunoglobulin receptor, for which copy number have been shown to be involved in early aspect of HDV control. So it is needed to further investigate if these CNVs on that locus could help us to understand the different degrees of susceptibility and resistance to HIV diseases in Tunisia and also in North Africa and the Middle East. We also noticed an increased burden of genes involved in metabolic pathways of medical relevance, such as the starch and sucrose metabolism, the carbohydrate digestion and absorption, as well as biosynthesis of unsaturated fatty acids. And among the genes involved in such a pathway, we have the amylase gene, the salivary AMY1 gene, and the pancreatic AMY2 genes, for which copy number changes are determinant metabolic states such as obesity, insulin resistance, and body mass index. And such markers are present in diabetes, which is also a real health burden in North African populations, as it shows the highest prevalence. So it is also preferable to investigate whether the amylase genes are associated to diabetes in addition to genetic predisposing factors already described and identified in Arabs. In addition, there is another gene involved in such metabolic pathway, and this gene is the MGAM gene, which is associated to a non-syndromic oral cleft, which is a birth congenital malformation. And also it will be preferable to investigate whether the deletions of this gene could be used as a biomarker for this congenital birth defect. We also investigate if these genic CNVs were involved in complex diseases. So we queried the genetic association database, which is an archive of genetic association studies on complex diseases. We identified 255 diseases that were clustered in 10 disease significant class. And among this disease class, we have the hematological diseases, the cardiovascular diseases, metabolic immune diseases, neurological diseases, and also CNV genes seem also involved in ophthalmological diseases such as vision and also in aging. We also queried the OMIM databases, and OMIM is the reference and the central databases of genetic diseases in human and their responsible genes. So we found that 103 RefSeq genes harboring CNVs are associated to 155 Mendelian diseases and phenotypes. The classification of these diseases according to the World Health Organization International Classification of Diseases showed that the most frequent diseases were the diseases of the nervous systems, the congenital malformation and chromosomal aberration, as well as the endocrine, nutritional, and metabolic diseases. Mental diseases and neoplasm were also prevalent. So we also classified the genetic diseases according to the transmission mode, and we found that 42% of these diseases were autosomal recessive diseases. So these findings show that and suggest that the Tunisian population is at risk of developing congenital malformation, metabolic diseases, and diseases of the nervous system, if we took into consideration our previous results on genetic diseases in our population. So we discarded also all intronic events, and we focused only on functional deletion and duplication that could interrupt the translation frame of a gene and could also affect the dosage of the gene. So we identify 63 disease genes harboring more than 50 CNVs. We split the disease genes in two groups. The first group involve genes with known CNVs, and the second group involve genes overlapped by novel CNVs that we previously shown. So in the first group, we have 56 genes affected by 53 CNV segments, and their frequency ranged from 1% to completely polymorphic. We recall that our individuals are healthy and free from any genomic disorders at the moment of the recruitment. And unfortunately, we don't have recent clinical data to confirm or not the presence of any genetic diseases. So in these slides, I'll show some striking findings on CNVs that affect the partial gene or the entire gene. So here we identify 6 deletions that affect only a part of a gene, and among these deletions, they affect genes involved in autosomal recessive disorders such as COX-10, STRC, DPY19L2, and FRSL1. And their frequency ranged from singleton to polymorphic and reaching 36%. So all these deletions are heterozygous deletions, and because they affect the autosomal recessive genes, the corresponding individuals that don't express the disease. We also identified 6 duplications that affect the complete gene. So the frequency ranged from 1% to more than 7%. They affect genes involved in autosomal dominant and autosomal recessive genes. And we don't know if these genes are actually dosage sensitive genes or not. And as I said, we don't have clinical data on these individuals to confirm the presence of any genetic disease. We also identified 3 deletions that affect the entire gene, affecting namely the CFHR3 gene, the UGT2B17, and the GYPB gene that seems highly frequent in our population. And it has been shown also in different studies that many genes could be deleted at the homozygous state, and these genes are qualified as non-essential genes and dispensable genes, as their absence in the human genome don't affect the phenotype. We also identified 5 mixed loci involving complete gene deletion and or duplication. And the striking example here is the PMP22 gene, which is a known dosage sensitive gene that is involved in the expression of Charcot-Marie tooth disease when it is duplicated, and another genetic diseases when it is deleted. So here, the deletion and the duplication of such a gene could probably lead to these diseases in these 3 individuals, but unfortunately we don't have clinical data. So, and we know that the corresponding individuals are in fact healthy at the moment of recruitment. We also investigate the tagability of the CNVs by neighboring SNPs in order to identify SNPs that could help to predict the deletion, because as we know, the genotype of the genotyping of SNPs is more easy than CNV genotyping. So we restricted our analysis to 223 B-allelic deletions identified in at least 4 individuals. As expected, all the B-allelic deletions had at least 1 neighboring SNP. The maximum SNP number in linkage disequilibrium was 15, with a median of 4, meaning that half of the B-allelic deletion could be tagged by 4 SNP or more, reaching to 15. And also we identified that only 47 B-allelic deletions were in strong correlation with at least 1 SNP. And this finding highlight and suggest also that the Affinimatrix SNP array is not well adapted to identify B-allelic deletion. We also found that the strength of the R-squared value decreases as the distance between the SNP and the CNV increases. The 111 tag SNP were annotated using RegulumDB, and RegulumDB is a database that annotate SNPs with known and predicted regulatory element in the intergenic region of the genome. So we found only 8 SNPs that with EQTL data and have also expression effects on the gene, as their score is 1F. And we also found 3 other putative functional SNPs, but without EQTL data and no effect on binding was reported. So among the 11 tag SNP here annotated with RegulumDB that are putative functional SNPs, we have 1 SNP that is associated to advanced age-related macular degeneration. And again, without clinical data, we could not confirm this phenotype in our sample. We also identified that only 22 B-allelic deletion tagged by SNPs are overlapping genes. And among these 4 B-allelic deletions, B-allelic deletions, 4 of them actually overlapped with omnin genes associated with the susceptibility to atypical hemolytic uremic syndrome, albinism, neuropathy, and mental retardation. And among these deletions, there is 1 deletion, CNP147, which is a 71 KB deletion that overlapped and caused the complete deletion of 2 genes, is in high linkage disequilibrium with SNP. The 3 other deletions are actually intronic deletions in 3 other genes associated with albinism and neuropathy and mental retardation. And this copy number variation or deletion, CNP147, is frequent in our population, as the heterozygous deletions is found at 44% and the homozygous deletion found at 14%. We also investigate the potential role of biallelic deletion in the etiology of complex diseases or traits. So we calculate the R-square value between the CNVs and SNPs that were available in the GWAS catalog. And we found biallelic deletion that were in perfect linkage disequilibrium with a SNP lying in the internal region of the gene CGCD, and it is this SNP is actually associated to multiple sclerosis. In addition, we found also two other deletions that were in high linkage disequilibrium with two other SNPs involved in and associated with metabolic traits like HDL cholesterol and triglyceride levels and obesity. In our recent work, we identify 218 CNV regions in 9 breast cancers from Tunisia that were negative for BRCA1 and BRCA2 gene mutations. And in order to further exclude CNVs that are unlikely to be associated with cancer, we compared our dataset, so the global CNV map, to the dataset generated on breast cancers patients. So we identified 58 segments that overlapped with our CNV map. And the majority of these segments were common and affect enriched pathways like olfactory receptor and xenobiotic metabolism. And the remaining 222 CNV regions were unique to breast cancer patients and are thought to contain CNVs associated with diseases given their rarity in the Tunisian population. So these results highlight the utility of the availability of such a CNV map and its application to medical genomics. So in order to give insight on population structure of the Tunisian population, we performed a comparative analysis of our data with data of the 1000 genome based on frequency values. And so we performed pairwise correlation, and we found a higher correlation between the frequency of our CNVs and the frequency of CNVs in the European population. And this correlation was less and weaker with the data of African samples. So this result suggests that the CNV profile of the Tunisian population is similar to the European profile and dissimilar to the African one. In addition, we performed a population stratification using copy number polymorphism loci with, and we performed a principal component analysis with the genotype of a CNP in our population compared to genotypes of CNP of 11 population from the HapMap project. We identified 3 clusters, an Asian cluster, an African cluster, and a third cluster in which the Tunisian population clusters with European samples. So this result is in agreement with previous studies using different kinds of markers that show the heterogeneity of the Tunisian population and the contribution of the European in its genetic background. So in summary, we provide the first genome-wide study of CNV in the Tunisian population in the absence of a Tunisian genome sequence. We also contribute to shedding light on this unstudied kind of variation. CNVs affected genes here reported are involved in biological pathway relevant to public health. We also provide the first LD assessment between the allelic deletions and SNPs allowing their imputation in future studies. So CNVs reported here should deserve further characterization and should be systematically investigated to assess significance and association with relevant diseases or traits. We also hope that our study will help to design a population-specific clinical array and help to interpret and assign the pathogenicity of such variation in our population. So we give the first insights of the CNV variant in the Tunisian population and raise many questions regarding CNV contribution to health issues in the Tunisian population and in North African populations, thus paving the way to the precision medicine implementation in North Africa and the Middle East. And thank you for your attention and patience. Thank you very much.